Hello and welcome to MarketOpen Direct Connect, a webinar series for companies to tell their story, why it's compelling, and why it's an opportunity to invest. I'm your host, Stuart Walters, and we're joined here today by Dr. Liz Dallimore, the CEO and Managing Director of Argenica Therapeutics, ASX code AGN. Liz will discuss the company's latest investor presentation, highlighting what success will look like in the phase 2 clinical trial, and will also participate in a Q&A. You can submit your questions through the Q&A tab at the bottom of your screen. In that, a recording of the webinar will be available on MarketOpen and Argenica's communication channels within the next 24 hours. I'd like to introduce our guest, Liz. Welcome to Direct Connect, and over to you.
So much, Stuart, for having me, and welcome to everyone online. I really appreciate you joining me. I really wanted the opportunity to be able to put a bit more colour to the investor presentation that we put out last Monday, a week ago, really talking around the what the sort of outcomes of our phase 2 clinical trial will look like. So I'll just jump straight in and for those of you that are new to the Argenica story, just to give a little bit of background first to sort of set the scene. So we are a clinical stage drug development company developing neuroprotective drugs that treat brain injury. So when we talk about neuroprotective drugs, it's really drugs that can protect brain cells from injury and from subsequently dying following that injury. So our lead drug candidate is known as ARG-007. What is really unique about ARG-007 is it is plurifunctional in terms of its mechanism of action, meaning that it works on a number of different pathways in the brain to really protect brain cells following injury. We have quite an extensive patent portfolio around this lead drug candidate, as well as other types of peptide drugs as well. And we have significant amounts of preclinical data, which has culminated in over 25 peer-reviewed publications. So really what we are trying to do and striving to do as a company is to change the lives of patients that suffer these horrendous brain injuries, such as stroke, traumatic brain injury, and a rare pediatric condition known as hypoxic ischemic encephalopathy. So this just shows you where we are on the preclinical and clinical pathway of our drug development program. So our lead indication is in ischemic stroke, and we are just nearing the end of our phase 2 clinical trial in that indication. We've also generated some really significant data in moderate to severe traumatic brain injury in preclinical studies, as well as in hypoxic ischemic encephalopathy. We are now sort of exploring a couple of other indications in sort of the mild traumatic brain injury and some neurodegenerative conditions as well. But really, a lot of this talk will be around that lead indication of ischemic stroke. So just to quickly touch on, I probably don't need to tell you that these are really huge unmet medical needs, which result in huge commercial opportunities for the company, because we are really trying to be the first company that has a drug that is able to protect brain cells after stroke, TBI, HIE. So these are large addressable markets, as you can see there. And just on the right-hand side of that slide, these are our preclinical results to date. So in stroke, it really was the work that we did in a non-human primate model that reflected what we are doing in the clinic that gave us the confidence to move into clinical trials in ischemic stroke. So in those studies, we saw a significant reduction in brain injury. Just quickly touching on where we are as a company. So as of the 31 March quarter, we had just under $13 million cash in the bank. That is sufficient cash runway to get us to the data readout for our phase 2 clinical trial and beyond. We're sitting around the 100 mil market cap at the moment of the company, and we've been really successful in attracting non-dilutive funding to progress some of our indications, which is fantastic. And as you'll see a little later on in the presentation, our trial is now fully enrolled and we've completed dosing in that trial. So I'll just give you a quick background to ischemic stroke. So ischemic stroke is caused by a clot in a blood vessel in the brain, as opposed to a hemorrhagic stroke, which is caused by a bleed on the brain. Ischemic strokes make up the vast majority of strokes. Around 85% of all strokes are ischemic strokes. A stroke in the US happens every 45 seconds. In Australia, every 11 minutes, someone will have a stroke. So, this is a huge problem that's getting worse and worse. So, the prevalence of stroke globally is going up. And of those people that suffer a stroke, only around 10% will make a full recovery. And the reason for that is because as soon as you have a stroke, as soon as you have that clot lodged in a blood vessel in the brain cells start dying almost immediately. And we know that around 1.9 brain cells die every minute following a stroke. So you get this proliferation out of brain injury in ischemic stroke. And that's really what Argenica is trying to address, to stop that brain injury that we see in ischemic stroke. So we're aiming to be a first-in-class drug addressing obviously this huge unmet medical need. So what is this opportunity in stroke? Currently, there is only one drug approved for stroke, and that is a drug that aims to dissolve that clot that is lodged in the blood vessel in the brain. These drugs are known as thrombolytic drugs. They can only be used in around 9% of ischemic stroke patients, and that's because they have a defined time window in which they can be used. But in that 9% of ischemic stroke patients, that global market currently is sitting around 1.1 billion. I think it's actually closer now in 2025 to about $1.8 billion globally, just to address that 9% of ischemic stroke patients. And due to the nature of the prevalence of stroke going up, that is due to increase to around 3.8 billion. So we are going after a market that's more like 30% of ischemic stroke. So you can just see how huge this opportunity is for Argenica in getting this drug to market. So I'll just quickly touch on the data that we do have to date before I launch into what we've been doing in the phase 2 clinical trial. So as I said before, the thing that really gave us conviction around taking this drug into the clinic was our non-human primate data. In this study, we tried to mimic exactly what we see in humans and the clinical care that humans get. So from suffering a stroke, administering our drug, and then removing that occlusion by a procedure known as a thrombectomy. So what we saw in this study was that our drug significantly reduced the amount of brain injury that we see on scans by 70%. So then that gave us the conviction, as I said, to take it into the clinic. There are a number of other things that we had to do to make sure that we really did have a compelling drug asset. The first to show that the drug doesn't actually exacerbate bleeding or increase hemorrhage. Ischemic stroke patients do have a propensity to have increased hemorrhage, so we needed to make sure that our drug didn't exacerbate that hemorrhage. We also needed to make sure that the drug could be administered with that clot dissolving drug, that thrombolytic that I spoke about before. Drug to drug interactions in clinical drug development is really important to understand. So we showed that it could be administered with that clot dissolving drug, and we'll also assess that further in this phase 2 clinical trial. And then we moved into a phase 1 clinical trial where we assessed the safety of the drug in healthy people. We saw no safety signals in that phase 1 trial, so that allowed us to move into the phase 2 clinical trial. So we have just completed dosing in that phase 2 clinical trial. For those not familiar with that trial design, I'll just quickly run through it. It is a blinded placebo, double-blinded placebo-controlled randomized trial. So what we were doing is patients were coming into the emergency department. They were getting screened for eligibility of to be admitted into our clinical trial. And then they either received a saline placebo IV infusion, or they received our drug at 0.3 milligrams per kilogram IV infusion as well. But pretty much everyone in the trial is blinded. So all the hospital staff in the emergency department, all the Argenica staff, we're completely blinded. So we don't know who's received the saline and we don't know who's received our drug. Then patients go straight into a procedure which is known as a thrombectomy. So this is where the clot sitting in a large vessel in the brain is aspirated out or sucked out of the brain. And really what we're looking at as the primary endpoint in this trial is safety. It is a safety study. It's really important in neurology drug development that we show that the drug does absolutely no harm to these patients. So the first thing that we really want to understand is to make sure that the drug doesn't obviously increase mortality rates, doesn't increase the number of adverse or serious adverse events that we would typically see in a stroke patient. And then the secondary endpoint is around infarct volume reduction. And I'll come to what that means, but this is really our efficacy endpoint. So, really, for the first time, as a proof of concept, understanding is our drug actually working in humans? We have fantastic data in animals and huge amounts of preclinical studies that we've done, but for the first time, really understanding as a proof of concept, does this drug work in humans? So as I said, the trial is now fully recruited. We've dosed all 92 patients that we needed in the trial, and the top line data is due in Q3 this calendar year. So we anticipate that will be later in Q3, sometime in September. So I just want to quickly touch on infarct volume reduction because this is where a couple of the new slides come into this investor presentation and give people a bit of flavour in terms of, I guess, does success look like in this clinical trial. So just these images are from a published paper, so not actually from our trial, but they really illustrate, I think, quite well what we mean by infarct volume reduction. Infarct of volume is just the volume of brain death that we see on imaging. So you can see here in these images, there is a blue section which we call the infarct core. So this white area that's color coded in blue is dead brain tissue, and there's nothing we can do about that dead brain tissue. Once it's dead, and there's no coming back. So that is the infarct core that we see. But you can see just to the left of that infarct core on the bottom images there, a sort of darker area which is color coded in that yellow shading. And that's what we call the penumbra, or the still salvageable brain tissue. So this is brain tissue that's sitting around the infarct core that is really vulnerable. So it's kind of sitting on the edge. It could kind of go either way. And this is really where we're hoping that our drug works. So what happens when these patients come in, we can see that they've got a certain amount of infarct core, but also, hoping they've got a certain amount of this penumbra for our drug to work on. These patients are infused with our drug or a placebo, and then they have this thrombectomy procedure that I mentioned before. But we know that around 70 to 80% of patients that have this thrombectomy procedure, that penumbra will turn to dead brain tissue. So you can see on the far right here that penumbra has turned white. So again, that's that sort of vulnerable brain tissue turning to infarct. So what we are hoping, best case scenario that we would see in our clinical trial, is that the patients that have the placebo, that saline placebo, they won't have anything there to protect the penumbra post thrombectomy. Whereas the patients on the treatment group that have received our drug will be in the system, circulating the blood, protecting those brain cells from dying, and that we won't get that yellow area turning to penumbra. So essentially then what we're measuring in the phase 2 trial is the difference at 48 hours between both of these images. So we'll look at the placebo group images and we'll look at the treatment group images, and we will measure the difference in that infarct volume, or that total brain cell death that we see on imaging. So what does that actually mean in terms of patient outcomes, and why are we measuring infarct volume reduction? Why is it important? There's been quite a number of peer-reviewed publications that have been published of late in the last couple of years that have now really articulated that reducing that infarct volume, that total amount of brain injury, is the strongest predictor of better outcomes for these stroke patients. And ultimately, when we come to moving this drug into later stage clinical trials, it is the functional outcomes that we will be measuring. We need to change the quality of life of these patients. So we do this by measuring on a scale. It's a disability scale. It's called the Modified Rankin Scale, or MRS. You can see it's quite a crude scale there. So 0, we've got no symptoms. 6, the patient is dead. And so we're sort of trying to see if there's a treatment effect to shift patients that have received our drug down that scale. So ultimately, really what we need to do in a later stage trial is move people down that scale. But we need to understand in this trial, well, how does the infarct volume reduction that we see, how does it actually then relate to this MRS scale? Because that will give us the confidence to know whether to move into that later stage trial. So this is what we come up with, and it's not us, it's not Argenica coming up with this. This has now been reasonably well established in the literature. So for the very first time, we have papers that have done a meta-analysis. So they've been able to take patient data from a whole raft of trials. So trials around thrombectomy, that procedure where they're sucking out the clot, which is a relatively new procedure. Looking at all the thrombectomy trials, because this is the first time that we've actually been able to record the level of infarct volume at either 24 or 48 hours and how that relates to that disability score, that MRS score that's typically done at 90 days. And so a paper by Liao et al., which was co-authored by Dr. Jeff Saver, who's the head of stroke at UCLA and does sit on our clinical advisory committee. They wanted to know, what is the minimal clinically important difference in infarct volume that we need to see to get an improved functional outcome for these patients? And typically what we're trying to do with MRS is ideally we're trying to move patients into the 0 to 2 MRS category, because this means that those patients can live a pretty much functionally independent life. So the level of disability they have is small enough that they can live an independent life. So that's essentially what we're hoping to achieve with these drugs. So again, looking back at this paper, what is that minimal amount, the smallest amount of infarct volume reduction that we do need to see? And what this paper showed is that really what we want to do is get at least one patient out of every 100 into the 0 to 2 MRS to live a functionally independent life. And so they calculated the infarct volume reduction needed to get one patient out of 100 into the 0 to 2 MRS is only 1.6% infarct volume reduction. They then looked to see if this was linear, and it is up to a point. So if you reduce the infarct volume by 5%, you get 5 patients out of every 100 moving into the 0 to 2 MRS. 11.5% results in 10, 17% results in 15, and so on. It is linear up to a point. There will be a point when obviously you just have too much infarct, too much dead brain tissue in that patient that you will not see an improvement in the MRS. And so what this paper does, what this big meta-analysis does, is that it shows us what that minimal amount is, which really is not that much. So what we're really trying to do in our phase 2 clinical trial is first and foremost is make sure there is a statistically significant difference between the saline placebo group and our treatment group. And if we do see that difference, what is going to give us comfort to move into a larger, later stage clinical trial? So the minimum that we really need to see is 1.6%. But I think as a company, if we could get that 5% difference, if we can go to clinicians, if we can go to pharma companies, if we can go to payers and say, we can increase the probability of 5 patients out of every 100 treated to live a disability-free life, that is huge. So I think that is really where we're sitting at it as a company, to say if we can show that, if we can show the probability because of what we're seeing on that infarct reduction will lead to this improvement in MRS, then we've got a really compelling story with ARG-007. And one thing I really wanted to point out before I look to wrap up for questions is that what we know is that even a small amount of infarct volume, or what clinicians have been telling us is that even a small amount of this infarct volume reduction will increase the chance a patient will walk, talk, and care for themselves, which is absolutely huge. And now, whilst it was anecdotal before, now we have the data to back that up, which is really exciting. Just to wrap up quickly on the stroke, I just wanted to highlight that we have submitted our investigational new drug application with the FDA, as well as our fast track application. So we are hoping to quickly have an open IND in early June, and then the fast track will hopefully be not too long after that. So I will leave it there. I'll just point out in terms of a couple of other catalysts that we've got coming up. We've got some TBI data, preclinical data for our next ferret study coming out in this quarter as well. So look out for that. TBI is a really exciting next indication that we've got some really compelling preclinical data on as well. So we're excited to see that. And obviously the big one is the phase 2 top-line data release in Q3 this calendar year. So thank you.
Liz, thank you. Fantastic presentation as always. We have had a few questions come in, so we'll get onto those right now. What statistical threshold will be considered a success for the primary efficacy endpoint around infarct volume reduction?
Yeah, so we look for a p-value of less than 0.05. If we're talking about that sort of statistical difference, that's the p-value that we're looking for.
And will Argenica apply for any adaptive trial designs or interim analysis approvals for phase 3 based on phase 2 data?
Yeah, so we in our pre-IND meeting with the FDA, we did have I guess they did agree with it was a proof of concept study. So this would never be a pivotal trial because we need to see functional outcomes. So it is very much that proof of concept trial that we're doing. Safety being absolutely key. I cannot, I don't think I can emphasize that enough in these sorts of trials. If we don't pass safety, then it's kind of a no-go type of decision. So that is the biggest hurdle that we do need to get over. But we can obviously use this infarct volume reduction now that we've got all these papers coming out around the correlation between infarct volume reduction and MRS at 90 days. We will be obviously going to the FDA in our sort of post-phase 2 meeting and talking through all the data that we've gathered. I will point out, we do have exploratory endpoints around functional outcomes that we are looking for. They're not powered in this study, so we don't expect to see anything, but it might throw out some trends that we would see in the trial as well. So, and then on phase 3 trial design, we are exploring all avenues at the moment. So we are looking at the potential of doing like a futility analysis in that phase 3 trial through that interim. We're looking at the possibility of adaptive trial design. The FDA is much more open to those sorts of trial designs in stroke. What the phase 2 trial is really doing is it's kind of really signal searching, right? So we've got patients coming in from time of stroke onset all the way out to 24 hours. And we've got sort of buckets of time within the trial. So a certain number of patients that might be, say, 0 to 6 hours, 6 to 12 hours, 12 to 24 hours. So if we think that the drug is working in a particular time frame, it might work better 0 to 12 hours, then we can adapt, obviously, the phase 3 trial to hone in on just those patients so that we can have obviously less numbers. We don't have to power with so many numbers to see a benefit.
Are there any biomarkers or imaging endpoints being tracked in addition to infarct volume and MRS that could support regulatory submissions?
Unfortunately, in stroke, it is really just the infarct volume. Look, we are working with a company that's got an AI tool where we are overlaying the AI tool around all the imaging. So there's a lot more granularity that the AI can actually pull out. So things like edema, hemorrhage, even measuring that penumbra. So we are doing that sort of stuff in the phase 2 trial. There's talk of blood biomarkers in stroke, but nothing that the FDA is kind of willing to accept at this stage. We're just even trying to get them to accept infarct volume as a potential sort of accelerated approval biomarker at this stage. But ultimately, it is in neurology, it is those functional outcomes that the FDA is looking for.
What's the breakdown of patients receiving ARG-007 versus placebo in terms of time to treatment, stroke severity, and comorbidities?
So we've tried to balance that out across the trial. So even though it's blinded, we're randomizing, but we're sort of randomizing them into to buckets as much as we can. So what we do know in the trial is that, so as I said, we've got that out to 24-hour time point, which is the first trial of its kind to go out that far for a neuroprotective drug. The reason we've done that is because thrombectomy standard of care changed out to 24 hours, and they found with thrombectomy that there was still a benefit those patients that had a good looking sort of penumbra that was still salvageable. So we have a few things in our trial that we are looking at. So when we patients are getting screened for the trial, we are looking at things like, the infarct core, how many sort of what's the extent of that brain injury. We apply what's called an aspect score. So any patients that have sort of extensive brain injury we've excluded because we need enough good sort of salvageable brain tissue, good penumbra. And then we've tried to, as I said, sort of put them in buckets. So we do know that I think there's only about 15 patients out of the 92 that are post the 12 hours. So that would sort of be a group on its own, and then reasonably good buckets within the sort of 0 to 6, numbers in the 0 to 6, 6 to 12 as well. Does that sort of answer the question? And then we've got the other main thing that we're stratifying in the trial is the TPA. So that thrombolytic use as well, because that's another really important question that we're trying to ask in the trial. So a good split between treatment, placebo with TPA as well.
How is the 1.6 reduction determined?
So it's determined through, so what's the clinically meaningful amount? So it looked at what's the total milliliter brain volume that needs to be saved to get at least one patient out of every 100 into a 0 to 2 MRS. So they determined that it was 0.6 milliliters reduction that they needed to see. So then taking the mean of the infarct, applying that reduction to get that percentage.
Do you think the drug has potential in other disorders of excitable tissues apart from the secondary penumbral injury?
Yeah, so it is an interesting question. So we do know the drug. We've got preclinical data to show that the drug is working on other brain cell types, so not just neurons, but it does have a positive impact on the astrocytes. We know that it reduces neuroinflammation as well. I mean, one thing we're really interested in is there a potential improvement in a safety signal with the patients that have had our drug? So in some of the preclinical studies, we do see sort of a trend towards a reduction in edema as well. So edema, which can lead to sort of intracranial pressure and that sort of stuff. So, we are again collecting all that information. It is very much a signal searching kind of trial. So we're sort of thrown it wide open to see where exactly is this drug working. And so, yeah, it's a really interesting question, right? Once we get that data out and we see, well, mechanistically, this drug is actually having an impact potentially on other areas.
You announced a new patient for surgically induced stroke prevention on May 22. Can you give us a feel for the market opportunity and size in this space as a treatment?
Yeah, so this is one that we've been having a few discussions with neurologists on. So it's a kind of a phenomenon that can happen with cardiac surgery, and there's certain cardiac surgeries. So TAVR being a sort of valve replacement surgery that patients have a 10% increased chance of having an ischemic stroke. So that's just when you're replacing the valve, something small breaks off and travels to the brain and causes a stroke. So it is the strokes tend to be smaller, so not these sort of large vessel occlusion strokes, but on the MRI imaging, you can see this infarct. So and the problem with these surgeries is the patient's obviously anaesthetized. So it's not until they come to that the surgeon realizes that they've actually suffered a stroke. So what we're looking at in this particular indication is the potential of giving our drug before surgery. So the pattern itself allows us to give the drug between um, 0.25 hours, so 15 minutes before surgery, all the way up to 4 hours before surgery. We know that the drug works for around 8 to 12 hours in hibernating brain cells, so plenty of time to sort of protect the brain. These patients are routinely then going in for brain scans, so if we're seeing that large clot, then that can be addressed, the clot can be removed, or a TPA can be given, and all the while the drug has protected the brain. So talking about their market size, if you're giving it almost like prophylactically, so you're giving it to, you know that it's safe, it could have the potential of protecting the brain during surgery from these surgically induced strokes. If you think about then the number of patients going in for cardiac surgery, that market is huge. But I will say, look, we don't have any preclinical data in that. We have done a global ischemia study it's called a 4VO. We did announce that very early on, back just after we listed. So we see that it is working in this sort of model. But really we wanted the US PTO had sort of knocked out that in our original patent, and we were very adamant that it should be included. So here we are, we've finally got it included as a new divisional patent, which is exciting, but huge market. But yeah, still a lot of work to do in working out where we might take that one.
If ischemic stroke could be diagnosed in the ambulance with new imaging technology currently in trials, would your drug work better if given then, just say 2 to 3 hours earlier?
Yeah, we think so. Obviously, the earlier the better, right? Because we know 1.9 million brain cells die every minute. So if we can give it earlier in the ambulance, then that would be great. So if we can utilize some of these, imaging technologies that are being trialed in ambulance at the moment, I think that would be really helpful. And the longer what we call kind of the dwell time, so the amount of time that the drug is circulating in the system before the thrombectomy, we believe it will have a better impact as well. So, yeah, we're keen to see some of these new in ambulance technologies get more prolific and into more ambulances for sure.
Step back to phase one. What effect did the drug have on the healthy volunteers? Did they go to sleep? Or if so, for how long?
No, nothing, no impact whatsoever. So, we interestingly, so the zero point 0.3 mg per kg that we've taken into the phase 2 clinical trial, that is our top dose that we tested in the phase 1 healthy trial. And that top dose in the phase 1 had absolutely no adverse events whatsoever. The only adverse events that we see that can potentially be related to the drug is sometimes a drop in blood pressure. You can kind of get that through just administering an IV anyway. But the main sorts of things that we saw in the trial was headaches, things like that. We actually saw more of those adverse events in the placebo group than we did in the treatment group. So it doesn't. So in terms of the way that the drug works, is it's when it's sort of getting into the brain, it's kind of shutting down these little channels on the brain cells that cause when you have a stroke. You get calcium rushing into your brain cells. So it's stopping that. But in healthy people, it's not going to have an impact because you don't have that high levels of glutamate. You don't have, sort of calcium rushing into the brain. So we typically wouldn't have expected to see anything in that trial.
Another one here, just can you please explain the difference in the assets of AGN and NYR and their approach to stroke?
Yes, look, I can't. I can talk at length about the mechanism of action of ARG-007. I know a little bit about Mirata's asset. So both are targeting calcium influx into neurons. Our drug is targeting that very acute phase, so the first rush of calcium that comes in through NMDA receptors and receptors. As I understand, the targets that MYR is targeting, which is still really, really valid targets, is that secondary phase of calcium influx as well. So, yeah, it's interesting because when I sort of talk to clinicians both here in the US, there is a view that neuroprotection will be a bit like oncology in the future, that there'll be, it'll be, kind of like a chemotherapy. There'll be probably a cocktail of drugs all working in different sort of stages. So ours works very much on the acute calcium influx. You'll have others that work on inflammation, others like MYR that sort of work on that secondary influx, and so could be really synergistic to ARG-007, so and then depending on really that sort of patient population as well, so that you're targeting. So different targets, but yeah, could definitely be synergistic in the future for sure.
Are there any plans to run subgroup analysis, i.e. age, gender, stroke severity, to identify potential patient populations for targeted use?
Yes, absolutely. So we will do a lot of ad hoc analysis. Again, as I said, it's really signal searching. So we are collecting huge amounts of data. We've got the AI tool as well that we're using. We've got a number of covariates, obviously, in our statistical analysis as well that we will account for. But absolutely, we will do that subgroup analysis and look for, is it working in a particular time frame, maybe a particular age, maybe particular sort of comorbidities. We are collecting all that data to really set ourselves up to design the best phase 3 trial moving forward.
Bear with me on this one. Is it necessary to know if a stroke is ischemic versus hemorrhagic to use ARG-007? And if so, why?
Yeah, we don't think so. So we have done studies in animals on hemorrhagic stroke, and the main thing is to make sure that our drug doesn't exacerbate bleeding in a hemorrhagic stroke patient. So the only drug, as I said, that is approved is a thrombolytic drug. TPA or TNK are the two thrombolytic drugs that are approved for ischemic stroke. These drugs do have a propensity to cause hemorrhage. So if you were to give TPA in the ambulance before you knew exactly what type of stroke it is, it could potentially be fatal to a hemorrhagic stroke patient because they can bleed out. Our drug doesn't work on the vasculature at all. So when we do studies in hemorrhagic stroke, we don't see that it exacerbates bleeding. So we can give our drug in the ambulance, regardless of if it's ischemic or hemorrhagic stroke. It was, I guess, really when we were listing the company and talking about the potential in ambulance use, really is still where we want to be. We see this drug really as being, if it could be in every single ambulance, and depending on what type of, patients coming in, they might have suffered a stroke, they might have been in a motor vehicle accident and had a TBI. They might have had a cardiac arrest and have a reduction in blood flow to the brain, which is causing brain injury. There's a whole raft of things. It's almost like seen as a kind of that wonder drug. So any time that a patient is potentially having an exacerbated brain injury, give ARG-007, hibernate the brain and buy that patient time to get into the hospital to get the treatments that they need. Running in ambulance trials are very, very difficult. So we made a decision as a company that that's something that we will do later down the track. Designing a trial in large vessel occlusion patients that are eligible for thrombectomy is really the best trial design that we can have to give us the best chance of seeing efficacy and getting the drug on market to that patient population as soon as possible. Once we have the safety in stroke patients, once we have, the drug on the market, then we can start expanding the use as well.
Liz, thank you. That's all that's come through today. Some great questions from attendees. So thank you for sending those in and appreciate you taking part in this Q&A. Fantastic presentation earlier. You're doing some great things in the medical space. So thank you so much for everything that you're doing. For more information about Argenica Therapeutics, head to the company website, argenica.com.au, or you can follow the company's social media channels or join their investor center via their website. I'd like to thank everybody for participation today. Wish you all the very best and look forward to chatting again soon. Once again, Liz, thank you very much for taking part in today's webinar.
Thanks, Stuart. Thanks. And thanks everyone for listening in.